Okay, so this video is part 3 of our protein work that we're looking at. What we're going to look at specifically today are the reactions, particularly that are required to break down proteins, but we'll also review obviously the reactions required to build up a protein, which we've looked at in terms of amino acids and the condensation reactions that are made there. But we'll also look at the opposite of that as well. So in terms of the study design, the concept here is metabolizing. So talk about what metabolizing really means, and we're going to look at that concept of metabolism or breaking down larger biomolecules for all of our food molecules that we look at. The proteins are just the first one that we're looking at. What we also will start to link to a little bit today is enzymes, and I'm going to talk a lot more about enzymes in the next video as well. I'll put this section of the study design in the next video as well, but you can clearly see, so remember our crossed out sections are the amended sections for the 2020 VCAR curriculum. So this, nothing really in terms of protein, but sections of the enzyme concepts that we will not be studying, and therefore we need to kind of, I suppose, not do those sorts of questions when we come across. Well, no need to do those questions anyway. Okay, so just in terms of general background concept, when we talk about macromolecules or macronutrients, depending on the term really you want to use, we're talking about carbohydrates, fats, and proteins, predominantly our three major macro food groups. So we know we eat these for lots of different reasons, particularly to carry out general bodily functions. Each of them have various functions in the body. We've talked a lot about, or we talked a bit in the last video about proteins in terms of their function around transport and growth and all sorts of other functions. But effectively, they actually are not really responsible for energy source in the body. If a human body starts to use proteins as an energy source, generally that's only going to happen if you're in starvation. Okay, because evidently what happens over a long period of time of burning proteins is your body enters what's called ketosis, which is very, very dangerous and produces some very nasty molecules in the body that are very dangerous to the brain. So the proteins are not actually used in the body predominantly for energy sources. In a sense, as they're broken down, they can be used for components of energy source, but if you have carbohydrates and fats in your diet, then they're going to be the primary energy source that the body uses. So when we eat these large macronutrients, and we've talked about this concept of large protein molecule, the digestive system of the body needs to break these large molecules down. Okay, the only way to actually get these molecules into the body and into particularly the bloodstream, and therefore able to be carried around to the cells of the body, is to break them down into much, much, much smaller molecules, or effectively break them down into what we started with. We're going to break them back down into the amino acid subunits that we started with. And the size of an amino acid means that the amino acids can actually be transported across the small intestine membrane and actually be enter the human bloodstream. Okay, so a couple of key terms just to start with. All right, we have the concept of metabolism, and I explained that before in terms of breaking down. It's a chemical process that breaks down larger molecules into smaller molecules for those molecules to then undergo digestion. A lot of you would be familiar with that word, but in terms of scientifically, digestion is effectively when our large molecules are broken down and absorbed by the small intestines in the bloodstream. That's where all of your most of your macronutrients gets absorbed in through the small intestine, and very little through other parts of the digestive system. Two types of reactions we're going to talk a lot about: hydrolysis and condensation. Now, we've already spoken about condensation because that's the type of reaction required to form larger molecules. So when we build up our amino acids, amino acid, that amide reaction caused the displacement of a hydroxide and a hydrogen ion, which means we get a byproduct of water. So it would make sense that if we're going to do the opposite, if we're going to, actually I might just break this down a little more. Get my pen right. We have your amino acids, right? We're building these up and build these up with amide bonds, and an amide bond water was cleaved off or produced off. Now, if you think about, we're going to do the opposite of this. Well, it would make sense then that water is required to be added or is required as an additional reactant for splitting our larger molecules into the smaller, or in this case, splitting a protein molecule back down to its smaller amino acid residues. Okay. So we're not going to go through the biological process of digestion. That's much more biological focused, and I think it's a little bit in VC biology. Once again, what we're looking at is chemistry, the reactions, and so on. I'll mention a few particular names of enzymes. They're not necessarily required for you to know these off by heart and what they do, but be very mindful of some of these key enzymes because they will pop up quite a lot, because there's some of the major ones that that can be certainly examined in terms of an analysis style question. So there's actually quite a number of enzymes that help digest protein. So we're talking about digestion, we're talking about breaking it down. And every single reaction of digestion requires an enzyme. All right, I'll talk about enzymes and how they work and what they are. Enzymes are actually effectively they are proteins, but they're a particular subgroup of proteins. I'll talk about that more in the next video. We're just thinking in terms of digestion. Digestion actually technically can start in the mouth, obviously if you're chewing something, but the digestion that occurs in the mouth is only ever going to be carbohydrate digestion. So protein digestion starts in the stomach, where an enzyme called pepsin operates fairly well at a pH of 1 to 2. And what it does is, you can sort of see here, you think about your large, more complex protein, and basically it starts to break it down, starts to digest it into just some smaller strands, okay? Not completely broken down into the subunit amino acids, but it starts the digestion. And it does so by acting on particular regions of the protein chain. However, to further break it down into peptides, into whether it's a single peptide, dipeptide, and so on, you need very, very specific enzymes for that. And I'll talk more about enzymes later on. This occurs, the generation of this sort of thing here, in the small intestine, okay? But evidently, the end process for this is that your protein is completely broken down or completely digested into single amino acids. All right, in its single peptide form, an amino acid can be absorbed from the small intestine through the intestinal lining into the bloodstream. Without complete digestion, you won't be able to absorb all those nutrients. And then some of you might be aware of actual diseases or disorders, sort of Crohn's disease and so on, that prevent people from this complete digestion of various macromolecules. All right, so chemically, what we need to be able to do is effectively, given a peptide or dipeptide, tripeptide, or a section of the protein, to be able to determine after hydrolysis where that hydrolytic reaction would occur. So that hydrolytic reaction is always going to occur at that peptide or amide link. All right, so that's what we're looking for if we're given a section of a protein. And we need to break that peptide link, reform our amide group, reform our carboxylic acid group into our two separate amino acids. Remember, hydrolysis must require water for this to occur. It also requires an enzyme. If you want to get a bit more specific with that now, that's great. Think about all these processes also require an enzyme to react, but also require water. Okay. So on this slide, what we're going to do is just practice that, just what I talked about before in terms of hydrolyzing or breaking down this. What is it? Looks like a tripeptide. So there's 3 amino acids here in sequence, and we're going to work out. You need to firstly find the amide bonds, and this one's fairly easy because these are fairly simple amino acids. I think we've got alanine, glycine, and alanine here. So their R groups are fairly simple. I mean, you'll have to do this given some more complex amino acids as well. So finding the amide functional group is going to be one of the easiest things. So you can probably see there's one here and then one here. So think about you're going to break the bond here and you're going to break the bond here. Okay, redrawing each of these, or all three of these amino acids then separately. Okay, so our first one, carbon, CH3, hydrogen, carbon, double bond oxygen, and complete that carboxylic functional group. You'll notice here that we've required 2 waters because we need one water for each bond that's broken, because we have a hydroxide here, and then in our amine group is where that additional hydrogen will be. Okay, now got to my carboxylic acid endpoint again, and then my third amino acid, which is the same as the one earlier. Okay, all 3 there, showing the tripeptide completely hydrolyzed in water. If you had to, you can work out what the names of these are. It's just showing by your 3-lettered symbol, alanine, glycine, alanine, using your data book. Remember, get used to using the data book and have it in front of you basically every lesson from now on. Okay. So there's another process that we're also going to talk about, which is called denaturation. Now that's very different from what we just spoke about. So previously, all that work we just did then, this is a chemical process in which an enzyme is required to break down a larger molecule. Okay, enzyme and water. All right, it's breaking it down into its smaller subunits. Technically, it could be rebuilt back up, and 100% what the body does. It often rebuilds proteins back up to suit the body's requirements. Denaturation is different. Denaturation still breaks down the molecule, but it particularly disrupts the structure of the protein. And what part of the protein it actually disrupts is the secondary, tertiary, and quaternary, if there is one, structures. All right, so it does not disrupt the primary structure. I'll explain why in a minute. But we know from our previous work that it's particularly this secondary and tertiary structure. We know from those intermolecular forces, we know that those structures are what folds our protein into a very specific shape. Okay. And if something disrupts these structures, what happens, I suppose the picture can kind of really, really simply put it there for you, is that if those structures are disrupted and altered in any way, evidently what happens is the protein kind of becomes what you can sort of say unfolded or unstructured, meaning that the overall shape can completely change or even just change slightly. Okay, what actually causes denaturation, what we're going to look at length, particularly when we look at enzymes, is temperature and pH, and it's temperature and pH outside the optimal range of the protein. All right, I'll break those 2 down in a minute, but it's those 2 processes that will cause denaturation in any kind of protein. All right, temperature and pH effectively change or disrupt in some way the intermolecular forces. Okay, but why then is it not a primary structure? Well, if I just draw my little amino acid sequence again, what bond is holding 2 amino acids together? An amide bond. Amide. An amide bond is covalent and it is intramolecular. It is strong. It is a very strong bond, much, much stronger than an intermolecular, hence why it requires an enzyme to actually break that bond. Okay, so denaturation will never completely destroy the protein into single amino acids. It will only disrupt, but as it does, it completely destroys the protein, but it will disrupt those secondary and tertiary structures specifically. Okay, so let's look at why temperature and why pH affects proteins. If you think about temperature and reaction rate to start with, as we know, if we increase the reaction rate of a reaction, if we increase the temperature, we will automatically increase the reaction rate of reaction. And the reactions that we're talking about here in living organisms are no different. Okay, this graph here is of particular importance to have a look at and look at the here. Okay, so yes, in a human body or in a living organism, we increase the temperature, increase the reaction rate. However, as you would be aware, living organisms cannot necessarily function in very varying temperatures, and this is predominantly due to proteins. So if we just think about the human body in terms of a 37-degree temperature optimal range, proteins have an optimal range of around 34 to maximum 40, and it will vary a little bit, but that's roughly, okay? The lower ranges will mean that the reaction rate, or if you think about reaction rate, think about the function of the protein, okay, will be a bit lower, a bit slower, and a bit diminished if particularly the temperature of the protein was around 34 or so. As you increase the temperature, you get to this obviously optimal temperature here. We would then argue in a human, it would be 37 degrees Celsius. So every protein in the human body, optimal temperature is that. What happens when it increases is we get a significant drop in protein function. All right, so think about, I like to think about the concept of like having the flu. That's quite relevant right now with everything going on. But if you've ever known, or if you've ever had or known anyone to have a really, really strong fever in which their temperature can be up to 38, 39, potentially 40, you would know that they are very, very sick. And effectively what's actually happening is their proteins in their body cannot function. They start to denature, they start to unravel, their structure completely gets destroyed, and therefore they cannot perform their function. If someone has a temperature of 39, 40 degrees, they should be in hospital, and would be in, I suppose, a lot of trouble if they couldn't bring that temperature right down. All right, so that's the concept we're talking about. Yes, lower temperatures do reduce protein function by reducing rate of reaction, but increasing the temperature above the optimal range, what that will do is that will disrupt our intermolecular forces. So think about this diagram down here. This is a little diagram that shows some disulfur bridges and some other intermolecular forces as well. These are disrupted by an increase in kinetic energy. So the molecules have so much kinetic energy that the overall intermolecular bonds just start to become disrupted and they break. They break and and effectively the structure unravels. All right, so the overall structure of the protein is completely gone, and therefore the function of the protein, or the protein effectively cannot function. Similarly, pH, or effectively the addition of an acid or the addition of a base, has a similar effect. Now, proteins can have quite varying optimal ranges for pH, and I'm going to talk about that in the next video. But if you think about that concept that we talked about in terms of azotarion, that we know that amino acids can behave as an acid or a base, depending on where they are in solution, but also particularly their R groups. The R groups can have, my pen back. They can have an acid or a base behaving functional group, can cause a problem. Because if you think about that, so you've effectively got an active acid or an active base in solution. And so depending on what kind of solution the amino acid or the protein is put in, these R groups can react, and they will react. So if you have an acidic R group, then the R group might react with a basic, will react, I should say, with a basic solution. If you have a basic R group or an alkaline R group, then that R group might react quite willingly and easily with an acidic solution. Now, if your R groups start to react, and therefore they're not interacting with one another. So what that means is the tertiary structure starts to become compromised, because it's the R groups that interact with each other at the tertiary level. If they're not interacting with each other, then the tertiary structure will start to change. All right. So depending on the pH of the solution and the amino acids and so on, it's a little bit more complex and a little bit more varied. But given a particular, you might be given a particular amino acid, so arginine. Arginine clearly has, an amino R group. So if this was put in particularly an acidic solution, you could argue that the R group then would interact with the acidic solution, potentially altering the overall tertiary structure of the molecule. All right. Okay. So continuing with denaturation, so the protein effectively chains become unfolded. What generally then can tend to happen is this process here, where, sorry, I'll just get my pen back, where denaturation occurs and then further kind of folding, but not the correct folding occurs, which is a process called coagulation. So the protein is in its proper shape here, okay? Temperature or pH causes denaturation, so causes the chains to unravel. But because the atom functional groups and everything else in part of the amino acid are still intact, they're going to actually continue to interact, and what you get is effectively the protein reforming a globular shape, but evidently the wrong one. So if the shape is wrong, the protein cannot function. The top picture is, it's just a real life example of this actually occurring. So the proteins in eggs, when you cook them, temperature affects denaturation or causes denaturation. And what you're actually going from is actual active protein here to no denaturation to the point of denaturation and effectively coagulation by that point. That's why the color changes. Okay. Okay, so the last bit, I'll just go in summary, a couple of little summary slides for you to use or look through. We've talked about amino acid sequence. We know that this amino acid sequence determines secondary and tertiary structure, and we've looked at what intermolecular forces are actually involved there. The secondary and tertiary structure therefore determines the overall 3D shape of the protein, and the overall 3D shape of the protein determines its functionality. Okay, now if we change, if we change temp or change pH here, that's going to stop the protein's function and basically also obviously unravel the protein, affecting the shape or affecting the secondary tertiary structure and the shape. The last slide here I've just put in as a review. Once again, we talked right back at the start around this concept of hydrolyzing larger molecules here. Okay, hydrolyzing those, adding in water into smaller molecules, and prior to that we talked about that condensation, the building up molecules. So you can see hopefully really clearly that it's effectively one reaction and then the other, and then the same reaction in reverse, whichever way you want to think about it. All right, that is it for today.